The primary advantage of our genotyping protocol is that it minimizes contamination with maternal DNA, which is the main challenge when genotyping formalin-fixed, paraffin-embedded molar tissues.
Flow cytometry is a fast and inexpensive method that greatly facilitates the diagnosis of Hydatidiform moles.
For each formalin-fixed paraffin-embedded, or FFPE, product of conception, prepare a 4-micron thick hematoxylin and eosin-stained section, as detailed in this video, for morphological evaluation by microscopy. Place the FFPE blocks on ice for 15 minutes to facilitate the sectioning. Adjust the microtome to cut sections that are 4 microns thick for microscopic morphological evaluation. Place the cold block in the microtome and cut one section from each block for H&E staining. Using forceps, transfer each section to a 45°C water bath. Pick up the section from the water bath with a positively charged slide previously labeled with the sample identification number using a pencil. Place the slides containing the sections in an oven at 65°C to allow the sections to adhere to the slides. Keep the slides in the oven for 25 minutes. Perform the H&E staining by submerging the slides into the appropriate staining jars for the correct time period, as tab